All right, guys, biochemistry. So in this one, we're gonna, pretty briefly go over nucleic acids, carbohydrates, lipids, proteins, and amino acids, and also some information about structure, uh, enzymes, and pathways. Okay, so that's a solid video. So let's see here. We got 5 questions. What are the monomers of proteins? What is the general term for the conversion of glucose into pyruvate? What are polymers? Which type of glycolysis has one glucose molecule of glucose become one molecule of ATP into NADPH? And hydroxyl is a polar group that forms what? Okay, and we'll go back to those questions again at the end where you can try to answer them for yourself after watching the video and getting some information here. So we'll start with nucleic acids. And as it says on the screen, nucleic acids contain a phosphate group, a 5-carbon sugar, and a nitrogenous base. Carbohydrates are sugars or starches that contain carbon, hydrogen, and oxygen. Monosaccharides are the most basic unit of carbohydrates. They are fundamental units of carbohydrates and cannot be further hydrolyzed to simpler compounds. Some monosaccharides have a sweet taste. Examples of monosaccharides include glucose, fructose, and galactose. Monosaccharides are the building block of disaccharides, such as sucrose and lactose, and also polysaccharides, such as cellulose and starch. Jumping to lipids, they contain long chains of hydrocarbons. Lipids consist of carbon, hydrogen, and oxygen. They are not soluble in water and are used for energy storage and insulation. Phospholipids. Here. Oh, sorry. Phospholipids form the membrane that make up the outer layer of all human cells. They play a key role in determining what enters and exits every cell. A steroid, and we need to know those. A steroid is an organic compound with 4 rings arranged in a specific molecular configuration. Examples include the dietary lipid cholesterol, the sex hormones estradiol and testosterone, and the anti-inflammatory drug dexamethasone. Steroids have 2 principal biological functions. Certain steroids, such as cholesterol, are important components of cell membranes, which alter membrane fluidity. And many steroids are signaling molecules which activate steroid hormone receptors. Okay, and then fatty acids are long strands of hydrocarbons and are used for long-term energy storage in the form of triglycerides. Okay, quite a bit on the screen here with proteins and amino acids. Proteins are composed of amino acids, which contain an amino group, a hydrogen molecule, and an R group. Amino acids are the monomers of proteins. Monosaccharides are carbohydrates composed of simple sugar ring monomers. When 2 monosaccharides undergo dehydration synthesis, they form a disaccharide. Amino acids are linked by a peptide bond that forms with the removal of a water molecule from the carboxyl add amino groups. Amino acids are the building blocks of proteins. There are 20 different amino acids that are in the human body. We can only synthesize 12 of them while we ingest the other 8. Hydroxyl is a polar group that forms hydrophilic amino acids, such as serine. Globular proteins are soluble in water, while fibrous proteins are not soluble in water and are found in instructional components like nails, which would be keratin, and connective tissue, such as collagen. Okay, with structures, we want to be familiar with all 4 of these, right? So primary structure is determined by the amino acid sequence of the chain. Secondary structure is determined by intermolecular bonding within the chain. Tertiary structure involves globular proteins, and the shape determines specificity. And quaternary structure means that there is more than a single chain. Okay, so those are all important, easily could show up on the test. Jumping to enzymes. Enzymes speed up the rate of reaction by lowering the activation energy, which is the energy required for the reaction to actually take place. Enzymes are formed by joining two amino acids, and enzymes are globular proteins. The molecules upon which enzymes may act, those are substrates, and the enzyme converts the substrates into different molecules known as products. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at rates fast enough to sustain life. Okay, so for the pathways, we have these three, and then we also want to mention glycolysis. So the Itner-Dudaroff pathway has one glucose molecule of glucose becoming one molecule of ATP and two of NADPH. The end product is two pyruvate molecules. The Emden-Meyerhof pathway has one molecule of glucose becoming two ATP and two NADH molecules, while the pentose phosphate pathway has one glucose molecule produce one ATP and two NADPH. So you can easily be tested on the differences between these three, and so you want to commit that to your memory. And then what we said, glycolysis. Glycolysis is the metabolic pathway that converts glucose into pyruvate and a hydrogen ion. The free energy released in this process is used to form the high energy molecules ATP and NADH. Glycolysis is a sequence of 10 enzyme catalyzed reactions. Most monosaccharides, including fructose and galactose, can be converted to one of these intermediates. And then we get back into our questions, right? And so this is a great time to hit that pause button and test yourself out.